Hello Bio30 and welcome to our video on abnormal meiosis, or what we call mutations. We are going to be focusing in on the differences of chromosomal mutations to point mutations. So we're going to start with chromosomal mutations. So today your job is to learn how to evaluate and demonstrate the consequences of mutations in all cells within our bodies, okay? Since we're talking about meiosis, we know that these are going to be sex cells. Your job is to be able to identify a karyotype and the criteria used to sort those out, as well as define nondisjunction, identify which stages of meiosis nondisjunction occurs, and define the words such as monosomy and trisomy. So we're going to start in the world of mutations during meiosis. So in your notes, we are right under the mutation section. We're going to talk about two types of mutations. One type is point mutations or gene mutations that happen during DNA replication. That's a completely different video. Today, we're going to focus on the chromosomal mutations which happen during meiosis. So although mutations actually occur quite frequently on a natural level, your genes, every time they replicate, get a little bit different, okay? Normally, we don't see those differences because we call them silent mutations, things that don't actually have a large effect. But then there are things that are causing mutations that we call mutagens. Mutagens are things that can significantly change your DNA or have a really big impact. So something like a physical mutagen would be UV radiation. UV radiation comes from the sun, and over time you can cause things like skin cancer. Skin cancer is a mutation of the cells on your skin, and then it causes the constant development of, like, for example, a benign tumor or carcinoma or something like that. All right. So the other ones would be chemicals. You can have things like benzenes that can have a huge effect, base analogs that look like nitrogen bases and get into our DNA. Heavy metals such as nickel, cadmium, they can have an effect. Bacteria can cause actually mutations to your genetics. And then you also have just what we call intercalating agents, and essentially they bind between nucleotides. So if we're talking about a chromosomal mutation, we want to know what that is. So a chromosomal mutation is a change to the structure of the actual chromosome. Us as humans have a diploid number, meaning our full set of chromosomes is 46. Okay, so we call that 2N or diploid being 46. If you are born with 45, 47, sometimes 48 chromosomes, these are what we would call chromosomal mutations because they are not the number that we should have in a diploid set of chromosomes. So how do we determine if something is a chromosomal mutation? Is we can use something called a karyotype. That's the picture you see here, okay? A karyotype is a picture of chromosomes, and we usually label it from the largest chromosome down to the smallest chromosome minus the sex chromosome. So chromosome 1 would be the largest chromosome we have, and then 22 would be the smallest, and then this is the sex chromosome. So this person here in this picture is male because they have an XY set of chromosomes. Now, in this case, you will notice that we only have one chromosome of a pair. So this is not meant to be an X. This is not showing a full chromosome. It's showing two small parts of the chromosome, okay? And so you can see here, down here, 22 pairs, okay? Means we have 44 pairs of chromosomes, 45, 46 with the X and Y. So again, there's the X and Y that shows sex determination. So why do we make a karyotype? The idea is that we want to determine if a fetus has either too many chromosomes, too few chromosomes, missing pieces of the chromosome, or extra pieces of the chromosome. So too many chromosomes, you're going to learn, is called trisomy. Too few is called monosomy. And then we don't really give names for the other ones, but for example, if you look at chromosome number 4 here, if you're actually missing a piece of chromosome number 4, you end up with a genetic disorder called Huntington's disease, which is a neurological disorder, and we've kind of talked about it a little bit, but essentially Huntington's disease will eventually kill you, and it develops later on in life, and that's due to a mutation of chromosome number 4. So how do we make a karyotype? What are the characteristics we're using? So we always want to look at the size of the chromosome. So when I make a karyotype, biggest to smallest. That's what I already said. So size of chromosome does matter, you guys. It's a big deal. We want to go biggest to smallest. We also want to look at how long they are. So not only the size, but how long they are is important because there's not much difference in size here, okay? But the length of their arm is going to be a little bit different. Also, we call it the centromere. Now, these are not attached, but if we had a full set that was X and an X, like if you can picture that, it's that piece in the center that matches. We want to know where those centromeres are. So you can kind of see there would be one here and here. You can kind of tell where they might attach based on where these are kind of naturally coming together. But the position of the centromere is important as well. And then the last thing that's really important is what we call banding pattern. You can see that there's light and dark spaces on these chromosomes. That's called the banding pattern. When they dye a chromosome, the dye that they use binds to certain sections of the DNA, so A's and T's, I believe it is, and it makes it darker than areas where there's C's and G's. So then you can look at the banding pattern to match it. So it's kind of like looking at what things are similar. So the steps to making a karyotype are pretty in-depth, but I'm going to simplify it down a little bit so that you have an idea. The first thing is you're going to collect a sample from the amniotic fluid. Now we've talked about amniocentesis and chorionic villi sampling. Essentially, really long needle, they're going to go in through the belly of the pregnant mom and remove fluid in the amniocentesis, the amniotic fluid. In chorionic villi sampling, they're actually going to take a little bit of the chorion. Now both of these contain fetal cells. So fetal skin cells, I should say. So what they do is they take it back to their lab and they need it to divide as many times as possible so they have a larger sample. Then what they do is they have to stop the division. You don't want to have all of these cells continually dividing because then you'll have too many to look at the chromosomes. So you take the dividing cells and you actually stop it and halt it in mitosis in the phase of metaphase. This is key. It has to be stopped in metaphase. And the reason it has to be stopped in metaphase is because at metaphase, the chromosomes are the most compact you're going to see. Remember, prophase is when they start to make those X's, okay? And then they line up at the equator. When they're lined up at the equator, they're in partners in the world of meiosis. They're homologous pairs. They're the most condensed that you will see. And so you can actually take them from metaphase and see them quite well. Once you've pulled and stopped it in metaphase and pulled the chromosomes, you're going to stain them with a special dye that shows that banding pattern. And then you're going to take a picture of it and analyze it. So you're going to take a picture and you're going to go and you're going to put it on a screen and you are going to start matching and counting until you have a picture like this. Now we're counting to make sure we have 46 chromosomes, 22 pairs, and an XY if it's a boy, or 23 homologous pairs if it's a female, XX. And we're checking to see if within that cell that we took all the chromosomes from, is there any extra chromosomes or is there missing chromosomes? Okay, so they arrange it by the criteria previously mentioned. Once they do that, they can see if they have a normal cell or an abnormal cell. An abnormal cell, what does that look like? So let's start to talk about a cell. So here are the cells in metaphase. They're lined up at the plate. Now normally, in metaphase, we would have 1/2 of each chromosome going to the opposite end of the cell. But you can see here, that's not going to happen. So the top cell will have 5 chromosomes and the bottom cell will end up with 3. Oops, just to show you. So the top one will have 1, 2, 3, 4, 5, and the bottom one will have 1, 2, 3. So abnormal meiosis is due to chromosomal abnormalities. And these are because the chromatids do not separate during meiosis. They stay together. Rather than the centromere being pulled apart during anaphase, they stay together. This process is called nondisjunction. That is really key, guys. Knowing that when it doesn't break apart in anaphase, it's called nondisjunction. Nondisjunction occurs in a couple ways. Okay, there's nondisjunction of anaphase 1, which essentially means homologous chromosomes move to the same pole. And then there's anaphase nondisjunction, anaphase 2 nondisjunction, essentially where the sister chromatids don't separate, which is what you saw in the last example with sister chromatids not separating. So let's talk it through here. Oops, sorry guys. Let's talk it through in this picture. So here I have nondisjunction about to occur. This is happening through anaphase 1. And how I know that is because my homologous pairs do not separate. Okay, so I have a cell, they're lined up at the plate. One is going this way, one is this way for the chromosome. This one does not. So non-disjunction occurs in meiosis 1 or in anaphase 1. So if we look at the cells that are developed, this one is lacking two chromosomes, and this one now has extra chromosomes. It's by 2 extra. So when they divide out of meiosis 2 down into their final set of gametes, let's say this is sperm, these 2 sperm are missing a chromosome, and these 2 sperm have an extra chromosome. So we say that they are n minus 1. N is representing 23 because in meiosis, you only create 23 chromosomes in every sex cell, half of the genetic material needed to make a full human. Remember, that's because the sperm and the egg will fertilize for 46 chromosomes. So your cells that are gametes need to only have 23. So this one is missing one. So we call this n minus 1 or 23 minus 1. So if this only has 22, but it doesn't, it only has 1, okay, but we're just pretending. And this one has 1 extra, so n plus 1. So this would be 23 plus 1. If we were using this specific example, this would be 2 minus 1 saying only 1 because there was 2 in, there was 2 here. This should have been n plus 1, so you have 3. Non-disjunction in anaphase 2 looks a little bit different. Non-disjunction in anaphase 2 means that the non-disjunction occurs in the second phase. So rather than all 4 cells having some sort of abnormality to it, only 2 cells do. So non-disjunction in meiosis 2 is actually a little bit better than non-disjunction in meiosis 1. So in meiosis 2, now we have one chromosome that is, one cell that is n plus 1, and one cell that is n minus 1. The other ones are unaffected. They are just haploid cells. So this is what it looks like when we talk about non-disjunction. So I talked it through. Now here is a little bit extra information. Non-disjunction during anaphase, and you can write this down in your notes, you have the space, is when 2 gametes have extra chromosomes and 2 gametes have 1 less chromosomes. It means all 4 cells are unhealthy. That's what non-disjunction in anaphase 1 does. Non-disjunction in anaphase 2 means that 1 gamete has an extra chromosome, 1 gamete has 1 less chromosome, meaning we have 2 unhealthy and 2 healthy. So making sure that we're distinguishing between the two to know that anaphase 1 and anaphase 2 non-disjunction, okay, can happen. But ultimately in the end, ladies and gentlemen, when you look at a karyotype, you cannot tell if non-disjunction occurred in anaphase 1 or if it occurred in anaphase 2. We just know that it can occur in either section. If you were to stop after producing a sperm and look at the sperm's chromosomes, you would be able to tell, or an egg's chromosomes, you would be able to tell if non-disjunction occurred in 1 or 2, but not after a full karyotype is completed. So non-disjunction, we say normal gametes have 23 chromosomes. Abnormal gametes form when we have either 24 or 22. So 1 more, 24, or 1 less, 22. To define the difference between 24 or 22, we call it trisomy. So trisomy means that when we look at a karyotype, one set of chromosomes is going to have 3 homologous chromosomes, or 47 in total. If we were to look at that, it would be this guy right here, 1 more 24. Because what happened is one of the cells, either egg or sperm, had an extra chromosome, resulting in a fertilized egg that had 3 homologous chromosomes somewhere. Oops. Trisomy 21, which means we have 3 chromosomes at the 21st section, the 21st chromosome, is called Down syndrome. And we're going to talk a little bit about that here shortly. Monosomy, 1 less, 22, is when we have a missing chromosome. So instead of having a full set of chromosomes and homologous pairs at each section, we're going to be missing one, resulting in 45 chromosomes instead of 46. Again, 1 less than 46 is 45. So again, it means either an egg or a sperm that only had 22 chromosomes came together to make a fertilized egg, fertilized zygote. Turner's syndrome is an example of a monosomy. So it's at chromosome number 23, which is the sex cell chromosomes, and it means that we're missing one of the sex cells. So here we have an X, and every male and female has an X, but we do not know if this fetus is meant to be genetically female by having an XX or genetically male by having an XY, because it's missing its second one. So what I'm going to do now is go through a couple quick examples of different trisomy and monosomy syndrome, examples, and just you can take notes as we go, or you can just listen. So here is Down syndrome, trisomy 21. That is what it's known as. And as you can see in the picture, there are 3 chromosomes at the 21st set of the pairs that they're supposed to have. There's now a triple, okay? This means they have 47 chromosomes in each cell. 1 in 600 babies have Down syndrome, and the incidences of Down syndrome actually increases as mother's age increases. So if you look it up, there's a correlation of what we call gestational, geriatric pregnancies, which essentially is a horrible way to say it because geriatric means elderly, but we're not saying elderly pregnancies, we're not saying elderly people getting pregnant, we're saying that the eggs of the female are considered older. And a woman is born with all the eggs they will have, okay? And as they age, the process of making healthy eggs gets harder. So as they get older, as they produce eggs, it may have some mutations in there. Okay. Also mutagens, being exposed to like x-rays and stuff like that while pregnant, can also increase chances. X-rays while developing sex cells and stuff, not pregnant. That's a different thing. Okay. Results in that. And this can result in a lower mental ability and a decreased lifespan. Kleinfelters Syndrome is when you have too many chromosomes in the 23rd pair. So rather than having XX or XY, you have a combination of XXY. Again, this is due to nondisjunction in the sperm and the egg, okay? And this is when a gamete contains 2 X's and then contains a Y, okay? Now, because of the Y being present, we learned that Y are determinant of testosterone and male characteristics. They are male at birth. However, although they look genetically they have male external genitalia, they produce female hormones. So if you look at the picture given, they can grow a beard, but they have narrower shoulders like females, wider hips like females. They can develop breasts. They tend to develop some chest hair, but very little. They tend to produce female pubic hair patterns because they have female hormones. And because of the female hormones, they don't develop properly in the external genitalia. So smaller testicle size, smaller penis, okay? So that is Kleinfelter syndrome. Turner syndrome is now looking at what we would call a monosomy example. This is an example of Turner syndrome where we have one X, but we don't have the other X, or we don't have a Y. So this is a female. They are born female because they have an X chromosome, okay? Again, this is due to nondisjunction. But what happens is that the X that contains genes for life, okay, we have them, but this child will stay a child, like looking like a child for a long time. They won't enter into puberty. So you will never, ever, ever have a pregnancy where there is no sex determination, that there's no XX or XY, because a cell cannot survive without an X chromosome. Jacob syndrome and triple X syndrome are again trisomy examples. Jacob syndrome is when instead of XXY, like Kleinfelters, we get XYY, and instead of triple syndrome, we get triple X's. Instead of XYY, we get triple X's. The first one, we get what we call the super male, and that's because of the 2 Y chromosomes. It has no real massive effect, except that they can have some learning difficulties, and they may produce extra testosterone, which may show in puberty, like excessive acne, more aggression, that sort of stuff. Triple X, it happens to 1 in every 1,000 females. The thing is that it delays language development. They may end up being taller, and it turns out that having an extra X may reduce a person's IQ by 10. The other ones we have are Patau syndrome and Edwards syndrome, which is trisomy 18 and trisomy 13. Unfortunately, if you have trisomy 18 or trisomy 13, there are way too many severe defects that although the baby may gestationally survive, meaning internally in mom and grow, they will not survive outside of the womb. So life expectancy for Edwards is about 10 weeks and Patau's is even shorter. They're born with non-functioning eyes with Patau's, cleft lips. For Edwards syndrome, it's even more deficiencies that they have. So unfortunately, these two are terminal. The last one I want to talk about, I believe, is the Fragile X syndrome, which is essentially instead of gaining or lacking chromosomes, this is an example of missing a piece of a chromosome. So this isn't trisomy or monosomy. This is just where on the X chromosome, there's a piece that gets pinched off at the bottom of the chromosome, causing this like indent, and it ends up being what we call Fragile X. It is inherited, it's an inherited form of intellectual disabilities, and it's an X-linked recessive trait. So women are carriers, and then usually boys that are born to women who are carriers can have Fragile X syndrome, okay? So that's it, ladies and gentlemen. Those are chromosomal mutations. Hopefully you were able to get everything in your notes, and I will talk to you soon.